Khateeb series in Human Anatomy and Physiology. Chapter 2, Cell Structure and Function. Episode 5. Cell Organelle Structures and Function. Part 2. In this part, we will continue covering the structure and function of the cell organelles. At the beginning, we're going to start with Golgi apparatus, which is often described as the post office of the cell. Located in the cytoplasm of eukaryotic cells, the Golgi is typically positioned near the endoplasmic reticulum and the nucleus. Functionally, Golgi is a critical organelle that serves as a molecular hub for modifying, sorting, and packaging proteins and lipids synthesized in endoplasmic reticulum and delivers them to final destinations. Let's examine the structure and polarity of Golgi apparatus. The structure of Golgi apparatus is made of a series of flattened, membrane-bound sacs known as cisternae, which are stacked upon one another. Inside each cisterna is the lumen. This lumen houses various enzymes and molecules that facilitate the essential biochemical reactions required for modifications of lipids and proteins. For proteins, these modifications include glycosylation, phosphorylation, sulfation, and proteolytic cleavage. For lipids, they include the addition of lipid groups, acetylation, and methylation. As shown in this image, the Golgi apparatus exhibits a remarkable structural polarity or faces. First, we can see the cis face, which is oriented towards the endoplasmic reticulum, and acts as the entry point for newly synthesized proteins and lipids. On the opposite side of the Golgi is the trans face that serves as the exit point for the products which are secreted as vesicles. In fact, the Golgi is involved in the formation of three distinct types of vesicles, each with a unique role. The first type is secretory vesicles, also known as granules, which primarily carry proteins and lipids destined for secretion out of the cell. These vesicles fuse with the cell membrane and release their contents into the extracellular space via a transport process called exocytosis. The second type of vesicle produced by the Golgi is membranous vesicles. These vesicles fuse with the cell membrane and contribute to replenishment and integrity of the cell membrane with lipids and proteins. The third type of vesicle involves the formation of lysosomes, which are intracellular digestive organelles. The Golgi helps in creating lysosomes by packaging hydrolytic enzymes into secretory vesicles. These vesicles then fuse with endosomes, a membrane-bound vesicles formed during endocytosis. The fusion of these vesicles creates the lysosome, which plays a crucial role in breaking down waste materials and cellular debris. Lysosomes are indispensable organelles within the cell, often described as the digestive system of the cell due to their critical role in intracellular degradation and recycling. In our previous discussion, we explored how lysosomes originate from the Golgi apparatus, forming as vesicles laden with potent digestive enzymes before being transported into the cytoplasm. Structurally, lysosomes exhibit a spherical or oval morphology and are encapsulated by a dual-layer phospholipid membrane. This membrane serves as a protective barrier that protects the cytosol and other organelles against their powerful hydrolytic enzymes. Within the lumen, an array of specialized digestive enzymes exist. These include proteases which digest proteins, lipases which digest lipid, nucleuses that digest nucleic acids, and glycosidases which digest carbohydrates. These enzymes work collectively to break down cellular debris and wastes, foreign substances, and damaged organelles into their fundamental building blocks. In addition, this enzymatic activity is finely tuned by the acidic microenvironment maintained within the lysosome, which equals pH of 5. The acidity within the lumen of the lysosomes is maintained by proton pumps that are embedded in its membrane. Also, the lysosomal membrane harbors a variety of transporter proteins, including cation transporters for sodium, potassium, and calcium, as well as nutrient transporters. These transporter proteins facilitate the movement of ions, amino acids, sugars, and lipids to the cytoplasm, ensuring homeostasis and biomolecule export. Now what about lysosomes' functions in the cells? In fact, lysosomes serve the cell with multitude of essential functions. These include digestion of pathogens, cell debris, and food particles, recycling of cellular components, and activating adaptive immune system. Let's first examine the role of lysosomes' digestion and immunity. As illustrated, once a bacterium or foreign particle is engulfed by a macrophage, it becomes enclosed within a membrane-bound vesicle known as a phagosome or phagocytosome. This phagosome subsequently fuses with a lysosome, forming a phagolysosome. Inside the phagolysosome, the engulfed material is subjected to enzymatic hydrolysis under the acidic conditions of the lysosomal lumen. Once digestion is complete, the breakdown products, such as amino acids, sugars, and lipids are recycled by the macrophage for cellular metabolism, while indigestible waste is expelled from the cell via exocytosis. Beyond degradation, lysosomes play a crucial role in immunity by allowing macrophages to act as antigen-presenting cells that activate adaptive immune system to fight infection. When a pathogen is digested by lysosomes inside the macrophages, specific antigenic fragments, such as peptides, are loaded onto major histocompatibility complex class II molecules which present the antigen on the macrophage surface. This antigen presentation alerts T cells, thereby triggering an adaptive immune response that mobilizes additional immune defenses to combat infection. Lysosomes are not exclusive to immune cells but also play a crucial role in nutrient digestion within unicellular organisms. In amoebas, for instance, food particles are engulfed via phagocytosis, forming a phagosome that subsequently fuses with lysosomes for enzymatic digestion. Like what happens in the macrophages as discussed before, the resulting nutrients such as amino acids, sugars, and fatty acids are then absorbed for cellular sustenance, while any waste is excreted through exocytosis. Apart from their degradative and immune functions, lysosomes also contribute to programmed cell death or apoptosis through a mechanism known as lysosomal membrane permeabilization or damage. When the lysosomal membrane is compromised, lysosomal enzymes, particularly cathepsins, are released into the cytoplasm, where they initiate apoptotic pathways. Various triggers, including low lysosomal pH and oxidative stress, can induce lysosomal membrane permeabilization. Once released, cathepsins activate the beta apoptotic proteins and degrade the anti-apoptotic BCL2 proteins. This leads to the permeabilization of the mitochondria membrane and release of cytochrome C, which can activate caspases proteins that push the cell towards programmed cell death. Finally, lysosomes play a fundamental role in autophagy, the cell's self-degradative process that removes damaged organelles, misfolded proteins, and other unnecessary components to maintain cellular health. Autophagy is triggered when the cell senses stress, misfolded proteins, nutrient deprivation, or dysfunctional organelles. Hence, the cell initiates the formation of a membrane structure called a phagophore. This phagophore gradually expands to encircle the targeted material, forming a double-membraned autophagosome. The autophagosome then fuses with a lysosome, creating an autolysosome, where hydrolytic enzymes break down the engulfed material into its basic building blocks, such as amino acids, sugars, and lipids. These degraded products are subsequently transported back into the cytoplasm, where they can be reused for cellular repair and energy production. Now, let's move forward and explore another tiny yet powerful organelle inside our cells: the peroxisome. As appears in these images, peroxisomes are small, membrane-bound organelles found in virtually all eukaryotic cells. They are enclosed by a single lipid bilayer membrane, which surrounds a dense matrix. The matrix of the peroxisome is filled with specialized hydrolytic enzymes that play a crucial role in metabolism and detoxification. However, unlike mitochondria, peroxisomes do not have their own DNA. This means that all peroxisomal proteins must be imported from the cytoplasm. To facilitate this process, peroxisomes contain transport proteins in their membrane, allowing the exchange of molecules between their interior and the cytoplasm. I believe that one question in your minds now is what enzymes are found in peroxisomes and how they work to facilitate metabolism and detoxification. The first major group of enzymes in peroxisomes matrix are oxidases. These enzymes catalyze the oxidation of various substrates, such as fatty acids and amino acids, by transferring hydrogen atoms to oxygen. This reaction produces hydrogen peroxide, which is a highly reactive and potentially oxidant substance. For example, the enzyme acyl-CoA oxidase catalyzes the first step in the beta oxidation of very long-chain fatty acids. On the other hand, amino acid oxidase helps break down amino acids. Interestingly, in all eukaryotes, the peroxisome matrix contains another antioxidant enzyme known as catalase. The enzyme catalase quickly converts the harmful hydrogen peroxide into water and oxygen to prevent toxic buildup. However, some species like rodents contain a crystalline core in their peroxisomes. This crystalline core is a dense, protein-rich region primarily composed of aggregated enzymes, mainly urate oxidase or the uricase. This enzyme catalyzes the oxidation of uric acid, which is a product of nucleotide metabolisms. Hence, human cell peroxisomes lack the crystalline core and the enzyme urate oxidase, which causes the accumulation of uric acid and the development of gouty arthritis, a common joint disorder in human. Therefore, the major cause of gouty arthritis in human is the accumulation of uric acid due to lack of the enzyme urate oxidase in the peroxisome.